Recent innovations in biotechnology are moving towards trends like artificial intelligence, data analytics, and automation to optimize production. Consequently, there has been a rise in consumer biotech or biotechnology products targeted directly at customers instead of biopharma or healthcare businesses. In this series of videos, I will showcase the trending innovations and technologies in biotechnology. So let us start learning one by one. To start with, let us first learn about biosensors. Biosensors are analytical devices that convert a biological response into an electrical signal. Quintessentially, biosensors must be highly specific, independent of physical parameters such as pH and temperature, and should be reusable. There are various types of biosensors such as enzyme-based, tissue-based, immunosensors, DNA biosensors, thermal and piezoelectric biosensors have been deliberated here to highlight their indispensable applications in multitudinous fields. Biosensors started in the 1960s by the pioneers Clark and Lyons. Various types of biosensors being used are enzyme-based, tissue-based, immunosensors, DNA biosensors, and thermal and piezoelectric biosensors. The first enzyme-based sensor was reported by Updike and Hicks in 1967. Enzyme biosensors have been devised on immobilization methods, such as adsorption of enzymes by van der Waals forces, ionic bonding or covalent bonding. The commonly used enzymes for this purpose are oxidoreductases, polyphenol oxidases, peroxidases, and aminooxidases. The first microbe-based or cell-based sensor was actualized by Divi's. The tissues for tissue-based sensors arise from plant and animal sources. The analyte of interest can be an inhibitor or a substrate of these processes. Rechnitz developed the first tissue-based sensor for the determination of amino acid arginine. Organelle-based sensors were made using membranes, chloroplasts, mitochondria, and microsomes. Immunosensors were established on the fact that antibodies have high affinity towards their respective antigens. The DNA biosensors were devised on the property that single-strand nucleic acid molecule is able to recognize and bind to its complementary strand in a sample. The interaction is due to the formation of stable hydrogen bonds between the two nucleic acid strands. Magnetic biosensors are miniaturized biosensors detecting magnetic micro and nanoparticles in microfluidic channels using the magnetoresistance effect, have great potential in terms of sensitivity and size. Thermal biosensors or calorimetric biosensors are developed by assimilating biosensor materials as mentioned before into a physical transducer. Piezoelectric biosensors are of two types, the quartz crystal microbalance and the surface acoustic wave device. They are based on the measurement of changes in resonance frequency of a piezoelectric crystal due to mass changes on the crystal structure. Optical biosensors consist of a light source, as well as numerous optical components to generate a light beam with specific characteristics and to beeline this light to a modulating agent, a modified sensing head along with a photodetector. Green fluorescent protein is a type of biosensor that is user-friendly, easy to engineer, manipulate and transfer into cells. Subsequent autofluorescent protein variants and genetic fusion reporters have aided the development of genetically encoded biosensors. Biosensors have been applied in many fields, namely food industry, medical field, marine sector, etc., and they provide better stability and sensitivity as compared with the traditional methods. In the next video, let us know more about biosensors. Please leave your comments and suggestions in the comments box. Please also like, share and subscribe.